All right, hello everyone. Welcome to week 9 of our Biochem Weekly Reviews. So this week we're going to be talking a bit about metabolism, and then we're going to shift gears quickly and move on to carbohydrates. The carbohydrate chapter is really the precursor to all of the metabolic pathways that we're going to be learning for the rest of this semester. So after we learn about carbohydrates, we shift and we talk about glycolysis, we talk about gluconeogenesis, we talk about glycogenesis, which is the formation of glycogen. We talk about glycogenolysis, which is the destruction or the use of glycogen, the pentose phosphate pathway, the tricarboxylic acid cycle, and then finally the electron transport chain. So that's basically what's going to start basically tomorrow, because I believe you did carbohydrates yesterday. I might have that wrong because the storm might have changed things, but yeah. So I'm just going to go ahead and jump right in with number 1. Oh, excuse me. There we go. So why are our metabolic pathways regulated? Right, so this question I asked last week, but this week I'd like to go into a little bit more depth on the answer. And specifically, I'd like to talk about this figure that you guys see here. So, and I'm going to give some examples. So in real life, in our bodies right now, glycogen is a not glycogen, I'm sorry, glycolysis. Glycolysis is a catabolic process. We start with glucose, we form ATP, NADH, etc., plus ATP, and then we get smaller building blocks at the end, okay? Another example of this is glycogenolysis, and the reason, so glycogenolysis, like I said, is the use of stored glycogen, and it's breaking down our large glycogen store into something smaller that can be used. So that's also a catabolic process. So glycolysis and glycogenolysis. And you're going to be learning about these two pathways more later. I just want to make sure that I'm mentioning them to relate them. So these are following the black arrows down that you see. So E1 reacts to make A into B, B reacts to make C, C reacts to make D, and so on until you finally get to this product that I'm highlighting in yellow, okay? In order to do that, let's look at E3 and E6. So this kind of circle in the middle, what this is doing is it's saying, in order to activate the forward reaction, we're going to use E3, okay? On the other hand, over on the right side, so looking at the anabolic mode of these enzymes, this is something like gluconeogenesis, which is very similar to the reverse genesis, to the reverse of glycolysis, of glycolysis, this is also glycogenesis. And I'm going to apologize in advance. A lot of these names sound very similar, so I may stutter over one of them. I may say the wrong name and try to correct myself. Just, bear in mind what I write is going to be more accurate than what I say. So anyway, so these pathways, they go up. So they follow what are also the black arrows here, but are the pink arrows on the left side. And the reason they do that is because they take our product and they turn it into our reactants. So one of the things that really confused me when I first started biochem is why? If gluconeogenesis just makes glucose from pyruvate in this case, well, why don't we just use the pyruvate for whatever? Like glucose is only going to be used for the most part for glycolysis. So, and that's a very good question. The reason this is something like gluconeogenesis can be used to reform glucose, and glucose can pass the blood-brain barrier and give your brain the glucose that it needs for glycolysis. Whereas pyruvate, from what I remember, cannot. Things like that are the reason that we have these reverse pathways. Glycogenesis, so the formation of glycogen from your glucose stores. This is useful, obviously, because it says when we have too much glucose, we can store it. And then glycogenolysis, when we don't have enough glucose, and we need energy, we can use our glycogen stores instead. So those are kind of the reasons that we have these reactions. So once again, we're going to look at that enzyme 3, enzyme 6, that middle reaction. And as you guys can see here, now enzyme 3 is inhibited and enzyme 6 is activated, okay? So that's the exact opposite, right? And if you notice, we're using the exact same enzymes, I guess green. We're using E1 twice, right? We're using E1 for both the catabolic and the anabolic. We're using E2 for both the catabolic and anabolic, and every other one except enzymes 3 and enzyme 6, right? And the reason that that's important is because, I think I briefly mentioned this last week, but we're going to be talking about, that's a very poor drawing, we're going to be talking about glycolysis and gluconeogenesis, again, very poor underlining, in a little bit. And the key to them is glycolysis is 10 steps, and gluconeogenesis is 11 steps, okay? They have 7 steps in common, but reversed. So this is, sorry, let me highlight this because this is really important. So this is what's happening that we see here. They both use, and we'll do blue, they both use E1 for the reaction between A and B. They both use E2 for the reaction between B and C. They both use blank enzyme for the forward and reverse reaction of the same kind. The reason that they have a different number of steps is because in the case of gluconeogenesis, one of these right here that would be E6 turning, let's say, D into C, is broken up and it says D into some intermediate, which I'll call I, and then into C. So it's an extra step, but it's doing the same thing. And so that's why there are a different number of steps, but 7 of the steps are the exact same, but reverse. Okay? So this is why we need regulation, because if they weren't regulated, the reactions catalyzed by E1 are very, very close to 0 in terms of their delta G values. So delta G, I'll call it not prime because we're dealing with biological conditions, is close to 0, meaning that either, I'm going to use arrows for this, the forward or reverse reaction could take place. All right? So we need to keep one of them regulated so that they don't keep both take place and kind of just waste energy for nothing. Okay? So that's really the bulk of what we need to know for metabolism. That entire chapter is really centered around this idea of regulation, and it kind of ties the previous stuff that we've been talking about with inhibitors, with allosteric regulation in general, into these pathways. Now we're going to shift gears and talk about carbohydrates. So the reason we talk about carbohydrates is just so that we kind of have an idea of the general notation that we're using for glycolysis and the other pathways that we're going to be talking about. Actually, no, there are a few more things in metabolism that I forgot. I apologize. So what role does compartmentalization play in metabolism? So for y'all who may not know this word compartmentalization in terms of like what we're talking about here, I'm going to give an example, and I'm actually going to scroll down below this figure to the next figure. So the Tca cycle, or I should say not just the Tca cycle, the pyruvate dehydrogenation happens in the mitochondria. Then the Tca cycle in the mitochondria. Then the electron transport chain along the mitochondrial membrane, but still mitochondria, right? Why can Tca, the citric acid cycle and oxidative phosphorylation only occur here in the mitochondria? Okay, so there are 2 reasons that we use that we have various compartments, if we want to think of it like that, of our cell used for various things. The first is because I'm sure you guys have seen the metabolic map that's like that crazy figure where everything is connected everywhere, that's just really complicated. In that figure, your professor might have put a star on pyruvate and called it the most connected metabolite. If they didn't, then I'm going to introduce you to that concept very briefly. Pyruvate can go just about anywhere. It can help become the precursor to one of our amino acids. It can give us energy through oxidative phosphorylation. It can go back into glucose through gluconeogenesis. It can do a million things, and that's why it's called I said most common, it should be most connected, I apologize. Connected metabolite. It's connected to so many pathways. So compartmentalization helps because pyruvate says, when I'm in the mitochondria, I'm doing eventually oxidative phosphorylation. I'm going to go through citric acid to get there, but eventually my goal is to do oxidative phosphorylation. When it goes to blank organelle, I don't know exactly which organelles are which, I don't have those memorized, but when it goes to maybe the endoplasmic reticulum, it'll do a different pathway. And when it stays in the cytoplasm, it'll do a different pathway, and so on. Okay? And so compartmentalization helps kind of keep things organized. All right, that's the first reason. The second reason comes to this figure that I skipped here, right? And this is the one in your book that is probably like arguably the more important one. I think it's still very helpful to talk about that compartmentalization for organization though. And it helps us create these concentration gradients, and specifically it helps us create energy gradients from those concentration gradients. And what I mean by that is this is a part of oxidative phosphorylation. What happens is from our energy transfers, which again, we're going to cover that in, I think, week 12, maybe week 13, we create a pH gradient. So we have all of these protons on one side of this membrane, and then the other side has very little. So just based on what we know of diffusion, of osmosis, of all these concepts, these H+ molecules want to go down their gradient, right? It doesn't take energy to do that. And therefore, since we have a gradient, we have potential energy. All right? So let me repeat that to make sure that's clear. When you have a concentration gradient of some molecule, you can use that concentration gradient to power something else, and therefore that gradient is potential energy for our cell. And so when the electron transport chain loads all of these protons on one side of this membrane, it creates that concentration gradient, which creates our potential energy. Now that potential energy can be used with, as I'm sure you guys can see right down here below, ATP synthase. And this ATP synthase, what happens, and I'm going to go as little depth as possible, this traveling down, you guys can barely see the purple. I'm going to, maybe green will work? I'm not sure. Yeah, green works well. As you travel down this, you rotate ATP synthase, and that that powers the ADP plus inorganic phosphate to ATP reaction and gives us ATP through compartmentalization. Okay, so those are the two important aspects of compartmentalization I want to cover. Now, if I'm not mistaken, okay, yes, we are moving on to carbohydrates. Oh, okay, there we go. So within carbohydrates, one of the first things that you guys need to understand is the difference between an aldose and a ketose. And really, the ose part stays the same. So really, the first part, what does that mean? Okay, so I hope that OChem 1 and OChem 2, especially OChem 2, is kind of coming back to you all, because this ald part, you know, it's in formaldehyde, it's in, well, all aldehydes actually. Aldehyde, and then like in a ketose, kind of sounds like a ketone, doesn't it? And that's actually exactly right. An aldose has an aldehyde, and a ketose has a ketone. And so that's the carbonyl functional group is the difference, all right? They can each have varying numbers of carbon. They're each describing a monosaccharide, so that doesn't really affect anything. Lnd conformation we'll talk about shortly, but it doesn't matter. You can have an aldose or a d-aldose, and the presence of nitrogen doesn't really matter. Okay, all right, so here are two examples, or I should say one example of each. So, I forgot to erase that. Okay, so as you all can see here, we have glyceraldehyde and we have dihydroxyacetone. Which I'm just going to abbreviate DHA, if I'm just to be honest with you guys, it's a long name. Okay, so glyceraldehyde, let's talk about this. It's got these isomers, L and D, which we're going to talk about shortly. But as you guys can see, highlighted here is a carbon double bound to an oxygen. So that's the carbonyl part. That also has a hydrogen attached to it, which makes it an aldehyde. Just a quick recap for those of you who don't remember that from OChem. And then on the other hand, dihydroxyacetone, oh, I should say glyceraldehyde is an aldehyde, as I'm sure the name suggests. Dihydroxyacetone is a ketone. Excuse me. So we look at our carbonyl, once again, carbon double bound to oxygen, and we see it's got bonds only to carbon, okay? Oh, and these are single bonds. It makes it look like a double bond when I do that, I apologize. But since it's only got carbon double bound oxygen to other carbons, that makes it a ketone. And that, since we're dealing with a carbohydrate, makes it a ketose, okay? And same with glyceraldehyde, since it's an aldehyde, it's an aldose, all right? So now let's talk about Lnd isomers. Okay, yeah, this is coming next. This is gonna be a fun unit. Okay, so let's talk about Lnd. So I'm sure you all noticed that the difference between the L and the D is this part right here, So as you guys can see in this right here down below, which I also forgot to erase some highlighting from, so I'm going to do that real quick. As you all can see from this, you can have carbon chains that are a lot longer and still be, because this is an aldehyde up here. This is a ketose, or I'm sorry, an aldose into ketose, I should say. They can have a lot more carbons. So what we're really looking at is this in pink, the last chiral center, okay? So, and I do have a question about Lnd isomers later. I just want to introduce it. L isomer, D isomer. All right, so what do we look for? The last chiral center. If the hydroxide group is to the right, it is D. If the hydroxyl group is to the left, it is L. And I personally think of it in terms of the hydroxyl group because I think left goes with, the letter L, right? So that's how I think of it. If you want to think of it in terms of the hydrogen, you absolutely can. I think thinking of it in terms of the hydroxide is easier. Excuse me. So let's talk about numbering, okay? So what I mean by that is you can have, for example, I'm sure you guys have heard in glycolysis, you have glucose 6-phosphate. I'm sure you've also heard of fructose 1,6-bisphosphate, for example. 1,6-bisphosphate. Well, that's all well and good, but why is whatever in fructose, like whichever carbon it is, number 1, and why is that one number 6? Why is this one number 6? And that comes down to the numbering convention that we have. So starting with the aldose over here, aldoses are very simple. You start from the carbonyl, that's number 1, and you move on. So 1, 2, 3, 4, 5, 6. Easy. Okay. But you can't do that for a ketone, or a ketose, I should say, right? Because, well, that can't be position 1, because then is this position 0? Is that position 2? How does that work? It just really doesn't. And so what we do is we say, okay, so our carbonyl is right here. Which side is it closer to? That side will be number 1. Okay, so this is number 1, this is number 2, 3, 4, 5, and 6. Okay. If it's right in the middle, so dihydroxyacetone DHA, it is right in the middle. You can number it either way. You use the lower number. So if I was going to say we phosphorylate one of the carbons, I would say it's dihydroxyacetone 1-phosphate, just because you really don't need the number, but I would say 1-phosphate if I had to give it a number, because that gives the lowest number to every functional group. Okay. All right. So for the above structures, I'm sure you can see, draw the L and D isomers of each. So I mentioned to y'all that Lnd isomers is we look at the last chiral carbon, okay? And that's true. But what I didn't mention is the full story. And the reason that there's more to it is because these are not just Lnd isomers, these are enantiomers. Enantiomers, okay? So in order to be an enantiomer, and I know this is like OChem 1 from over like a year and a half ago, it's very hard to remember. So for it to be an enantiomer, it needs to be a non-superimposable mirror image. Okay, what does that mean? That's a lot of words. The way that I think of it and how I memorized it is every chiral center flips. All right, flips, okay? So again, what does that mean? So I'm going to come up here and I'm going to be using D-glucose as an example. So the reason that D-glucose is because, well, we go to the last carbon. Well, carbon number 6 is not chiral because it's got 2 hydrogens attached to it. Therefore, it doesn't have chirality, right? Because to be chiral, you have to have 4 unique substituents. All right, so we go up 1, and we're going to say, all right, let's look at carbon number 5. And in this case, it is carbon number 5, which is why it's highlighted in that pinkish color. Is it red? Pinkish reddish? I don't know. And we say, if it's to the right, if the OH, oh, you guys can barely see the highlighting. If the OH is to the right, it's D. I was going to say R and S. I apologize. If it was to the left, we would say L. Okay. But if I were to just draw this exact same thing again, carbon OH, H, carbon H. Sorry, give me one moment. If I were to literally copy this down verbatim, except just swap this around, this is not L-glucose. It's not. This is a different 6-carbon aldose. I believe this ends up being mannose, but I'm not 100% sure. I would need to look back at it, right? So I'm sure your professors in their slides showed a kind of huge figure of, 3-carbon, then 4-carbon, then 5-carbon, and 6-carbon aldoses and ketoses. Just for the record, I don't know which ones you have to memorize. I would ask your professor to confirm. Usually it's like glucose, fructose, maybe mannose or something like that, 1 or 2 more. You normally don't have to memorize more than 4 or 5. But the reason that I say this is not L-glucose is because of this concept right here. It has to be an enantiomer. Since it is an enantiomer, and every chiral center will be flipping, what's going to happen is we flip. Well, hang on. We said 6 is not chiral, and 1 isn't chiral either. 2 through 5 are. Okay. So in order to draw, sorry, I'm going to zoom out just a little bit so I have some room. In order to draw right there the L isomer of glucose, I need to say, okay, CHO, that stays the same because it's not chiral. Position number 2 is going to flip. So I have an H on the right and an OH on the left now. Position number 3 is also going to flip. So I have an OH on the right instead of the left. Position number 4 also flips, and so on. And then, I've run out of space. So what I'm going to do is I'm actually just going to erase the first one, and I'm just going to move the rest of this up a little bit for the sake of not having to waste your time redrawing. That's fine. Okay. All right. So this is, we'll say 2, 3, 4. Position number 5 is also going to flip. Okay. So let's start off by saying, okay, we can confirm this is indeed L-glucose because, well, the OH is on the left. Cool. All right. We can, or I should say, this is an L-aldose for now. We don't know which one it is, but just based on the fact that that's position number 5, and once again, I left off 1 and 6 for the sake of space because they don't change. In order to confirm that it's glucose, I now have to go and say, all right, let's compare position number 2. We have OH on the right. Over here, we have OH on the left. Okay, so that is the opposite of glucose, of D-glucose so far. Position number 3 is also the opposite. Position number 4, I accidentally drew the same way, so I need to flip that. So this is not, in fact, L-glucose. Position number 4 is going to be that. There we go. Now this is L-glucose. I didn't even realize I was making that mistake. So this is L-glucose because every chiral center has flipped. And I'm going to go ahead and swap that over L-glucose now. Glucose. There we go. All right. Because this is clearly not enough space, I'm going to go ahead and draw L-fructose beside regular fructose, or beside D-fructose, I should say. So the reason I called it regular, and I guess this is a great time to mention it now, is most of the sugars in our body are in their D form. I want to say almost all of them, but I don't know the exact number. I just know that it's pretty much like most of them. And so if I were to ask you, for example, what is it that our body needs, it would be it would be D-glucose, not L-glucose. Okay, COH, H, and this is CH2OH. Okay, I believe I flipped everything here. Yes, yes, and yes. Okay, so I know that this is an L-ketose now because of this, and well, this obviously makes it a ketose. I know that it's L-fructose because every chiral center flipped. Okay, I hope that makes sense. I apologize that I did take a little bit of time on it, but I really want to make sure to emphasize this because it can be confusing. Okay. All right. So, moving on. What is the difference between the alpha and beta forms of a sugar? Okay. Once again, there is a tiny little piece that I forgot to erase. I apologize. This is copied from my notes, and I thought I'd erased everything. Okay. So the alpha and beta forms of a sugar come from what's called the, I always get this mixed up, it's the Haworth projection. I believe that's correct. So the Haworth projection is the ring that we're familiar with, basically. And so let's start off by saying, okay, what ring is it? So you can have two different rings, and you'll see that, that's fine. Okay, I kind of spoiled it. That's fine. So you can have two different rings. You can have a pyranose, which I'm just going to draw up here, and pyranose, or you can have a furanose. Furanose. Okay. These tell you how many carbons are in the ring. So a pyranose is a 6-carbon ring. A furanose is a 5-carbon ring. Okay. So you may be asking, well, wouldn't every 6-carbon ketose or aldose form a pyranose and every 5-carbon form a furanose or something? It's not quite true. So I'm going to describe how this reaction can work, and then we're going to like how the folding happens, basically. And then we can go into a little bit of depth about why it happens the way that it does, or like the different forms it can take. Okay. So it's important to recognize that this is called cyclization. Okay. That's not that important, actually. I changed my mind. So let's talk about the reaction. The reaction relies on using the aldehyde or the ketone, or yeah, the aldehyde or the ketone. And we're specifically talking about the functional group, not the sugar here. What happens is an alcohol will attack this aldehyde. Okay. So which aldehyde does it? The answer is carbon number 5 in the case of glucose. Okay. So this hydroxyl group right here is going to come in and attack this carbon. When it does, let's see, I'm just going to draw the OH attached to the carbon. So that's this one. So this comes and attacks, that's the carbon. So this would be carbon 1, this would be carbon 2, and this is obviously the hydrogen. When that attacks with its lone pair, this comes up and gives this a negative charge. And then a bond forms between this oxygen and this carbon number 1 to form a 6-member ring. Okay, so let's draw that. So what I'm specifically going to draw is what's in the ring. Okay, and what's in the ring in this case is an oxygen, because an oxygen attacked, and so it's attached. The carbon number 1, carbon number 2, carbon number 3, number 4, and number 5. Number 6 did not get lost, but because we attacked with carbon number 5, carbon 6 is not a part of the ring. Instead, carbon number 6 is sticking out. Okay. Now you'll notice I didn't draw any of the functional groups, or like the hydroxides, or the hydroxyls, the hydrogens, anything. The reason I didn't is because their location matters. Okay. Just like whether the hydroxyl group is to the left or to the right in the, like in this linear diagram, if the hydroxyl is up or down, that makes a difference. That changes what we're looking at. And so going slowly, because I'm going to make a mistake if I don't, let's go ahead and label this. So I'm going to go ahead and draw the alpha configuration of this, and then we're going to talk about how it differs from the beta and why it differs from the beta. Okay, and this is D-glucose, so I'll be drawing alpha D-glucopyranose. So that's, I'm actually going to go ahead and write that because I think it's important. Alpha D-glucopyranose. Okay, so carbon number 1 has its alcohol pointing down, its hydrogen pointing up. I'm going to highlight this for a reason that we'll talk about in a moment. You probably already know if you took a intro bio class, but I'm going to do it, carefully anyways. Okay. Carbon number 2 has its hydroxyl group pointed down, and I'm not going to draw the hydrogens for the sake of space. Carbon 3 has its hydroxyl pointed up. Carbon 4 is pointed down. Carbon 5 doesn't, well, I mean, this is the hydroxyl of carbon 5, and carbon 6 is CH2OH. Okay, so let's look at this. Okay, so I hope you all can see my mouse well. So carbon number 1 points down. We're going to skip carbon number 1 since it's the weird one. Carbon number 2 points down. The hydroxyl group, the alcohol, is to the right. 3 points up, the group is to the left. 4 points down, group is to the right. 5 is the exception. Okay. In other words, over here, what's pointing to the right converts to down over here on the Haworth projection. Okay. So let's highlight these in blue. Carbon number 2, down. Right, down. Carbon number 4, right, is down. And we'll use green for carbon number 3, which has it up, and then 5 and 6 are the exceptions. Okay. So I highlighted this one in yellow. Why did I do that? And the reason I did that is because if you all remember, way back to organic chemistry, you can get what's called a racemic mixture when you do some reactions. A racemic mixture is where, for the sake of simplicity, I'm going to use OChem chirality notation. You can get both R and S isomers of your product. Something similar happens here. When you react this reaction right here, you can either get the equivalent of these blue highlights or the equivalent of the green highlight. And that's what you see down here. Alpha glucose and beta glucose differ in the position of the alcohol on the first carbon group. Okay? Let's repeat that one more time. Alpha glucose and beta glucose differ because of the position of the hydroxyl on the first carbon group. Okay? And you can get both. It's just you need to know which one you're dealing with and you need to be able to identify. Okay? Oh, we're not there yet. Okay. All right. Actually, I think we're just about there for now because if I remember correctly, yeah, okay, we've got a lot more. Okay, so a glycosidic bond. Let's talk about what a glycosidic bond is. A glycosidic bond is, well, glyco is kind of that word that we use for sugars, right? Glycogen, glycolysis, whatever. It's a bond between two sugars, basically. Okay, so, and I'm going to be using this example up here of alpha glucose and beta glucose for this bond, right? So the way that this works is, I don't want to say simple, it's not really that simple. It's fairly straightforward, even though it looks weird. I think that's the best way to put it, right? So what's going to happen is fairly similar to what happened originally. A hydroxyl group will come and attack the carbon. Right, and I'm drawing 1 and 4 because 1 and 4 is a very common kind of binding, it's a very common bond. A 1,4 glycosidic bond is very common. All right, so when it does that, the hydrogen, well, eventually, basically what's going to happen is this oxygen right here is going to be what connects. I just realized you guys can barely see since I'm using black on black. Let's switch to red. This oxygen is going to be what connects this carbon to this carbon. So the end result, and I'm just going to draw the stuff in immediate proximity for the sake of convenience. The way we draw it looks like that. All right, and that's CH2OH, and this is with an OH, and then obviously there's more stuff coming off of all that, but we don't really care for now. Okay, cool. So this is what's called an alpha 1,4 glycosidic bond. Acidic bond. All right, so let's talk about what's missing, right? So we have this oxygen here. Well, the hydrogens are the same. The hydrogen's up here. So that didn't leave, that didn't leave. This oxygen was originally attached to this hydrogen. So this hydrogen got dumped. That's one thing, right? But this other alcohol also left, right? So in other words, what do we get out of it? Water. Okay. D, excuse me, right? So I've explained alpha glycosidic bonds. Let's talk about beta glycosidic bonds. I also want to point out, alpha glucose forms alpha glycosidic bonds. It's just fairly simple, right? So let's talk about beta. So I'm going to use the right half of this beta glucose, and I'm going to draw another glucose molecule, or part of another glucose molecule, I should say, for the rest of it. And this is going to be literally the exact same as what we see like for carbon number 4. This one is going to have OH, this one is going to have H, CH2OH, and then it goes on, that's an oxygen, and that goes like that. And I don't really care to fill the rest of this in. You guys know they're there. Okay, so what I didn't do is I dropped the oxygen there, right? So if you notice, effectively the condensation reaction that happens, and I'm just going to clear some space here. The condensation reaction that happens is these two come together, the two alcohols, and they leave you with, to oxygen, oh, that was a terrible drawing, to this oxygen, to this carbon, right? The same thing happens on this side, except it's crooked now, right? So you're still going to get carbon to oxygen to carbon, but now it looks differently. And the reason it looks differently is, or I should say, you guys see that it looks differently. Let's look at how it actually appears. Okay, it's a little bit weird. It ends up being drawn like that. And then this is the CH2OH and so on. So this is a beta 1,4 glycosidic bond. All right. So the reason these alpha and beta bonds are very important is because, for example, the difference between a few disaccharides can be an alpha or a beta bond. So the examples given in your slides are sucrose, for example. Sucrose is always glucose alpha bonded to one alpha bond between positions 1 and 2 on fructose. Okay, so in this case, it's, oh, I guess I should have mentioned how to read these, shouldn't I? Because I kind of said this is what it is. I never explained why. Right? So the beta is the type of bond, right? This tells you it's a beta or an alpha bond, okay? Right? 1 comes from, this is carbon number 1, okay? Likewise, I guess I'll use blue. 4 comes from the fact that this is carbon number 4, right? And then glycosidic bond is just kind of there, right? So if I were to say this is a, well, we won't use glucose. I'll say glucose alpha 1, 2 fructose. What this tells me is it's an alpha bond, obviously, between carbon number 1 of glucose and carbon number 2 of fructose. And you take, you know, the first number goes to the first thing, the second number goes to the second, right? It's fairly straightforward there. So this tells me, and this is sucrose. This is the example that I was giving earlier. Sucrose is always glucose bound to fructose in an alpha linkage between the first carbon of glucose and the second carbon of fructose, always, right? So if it was a beta bond between the exact same carbons on the exact same saccharides, it would not be sucrose. Okay, that's very important, right? Okay, so tangent over, let's move on. What form is a naturally occurring molecule of glucose most likely to take? Great question. So the reason that we look so much at the D isomer is because the D isomer is almost always the more stable isomer. Okay. And so just right off the bat, I'm going to say it's going to be one of the D isomers. Okay. So now, is it going to be a 5-member ring or a 6-member ring? Obviously, in this case, since I just drew it for you all, you know it's a 6-member ring. Excuse me, it's a pyranose. However, the reason that this is the case is, and there are exceptions to this. So I want to mention fructose tends to form furanoses. All right, fructose tends to form furanoses. However, 6-member rings, as I'm sure you either remember or try to block out of your memory from organic chemistry, 6-member rings are more stable. Most stable, I guess would be an appropriate way to say it, because of bond angle. All right. So while there definitely are 5-member rings that are stable, like fructose, if you're really not sure, and like you don't know any exceptions, guessing that something is a pyranose is not wrong to do. Okay. All right. So, spoiled myself a little bit. Okay, if I have a disaccharide formed from a beta glycosidic bond between the first carbon of a glucose molecule and the fourth carbon of a galactose molecule, how should I name it? So right off the bat, I'm going to eliminate A and D because they don't reference the fact that it's a beta bond. This is very important. Okay, right. And then it's the first carbon of glucose and the fourth carbon of galactose. So for this, what I'm looking at is it goes glucose, and then 1 should be the first number, and it is. And then it goes 4, which means galactose needs to be the second number, right? So B works. C does not. C does it exactly backwards. It does, and I'll try to use the same highlighting, so galactose and 4, and then glucose is 1. It just does it backwards, right? So I think it's really important. Like, this is fairly intuitive, right? The first number goes with the first sugar, the second number goes with the second sugar. So yeah, okay. All right, so number 10. What is the difference between amylose and amylopectin? Actually, before we jump into this question, I think it's really important to talk about these two, not in general, but why we talk about these. So amylose and amylopectin are polysaccharides, okay? And specifically, they are they're polysaccharides that are found either like in real life, right? So either in the human body, in plants, in various whatever. So they're important because, for example, glycogen is a polysaccharide. So we like to talk a little bit about polysaccharides so that we understand them, so that when we approach glycogen, it makes more sense, okay? Just want to mention that because I think sometimes it can be really easy to just kind of disregard this unit or this section of the unit and just kind of not care about it, but it is important, okay? All right, so what is the difference between amylose and amylopectin? This is a fairly basic question. The answer to this is that amylopectin is a linear chain of glucose units that are alpha-1,4 linkages, okay? So amylose is linear, glucose alpha-1,4. And I apologize for my handwriting, it is very bad. Glucose, there we go. All right, amylopectin, on the other hand, is a branched chain in which branches are created with alpha-1,6 linkages, okay? So branched, so I think of amylose as amylopectin, or I'm sorry, I have that backwards. I think of amylopectin as amylose plus something. So amylose plus branches, alpha-1,6, made by alpha-1,6 bonds, okay? So amylose is made of alpha-1,4, amylopectin is made of alpha-1,6. That's incorrect because amylopectin has both 1,4 and 1,6, okay? Amylose is branched, amylopectin is linear. That's the exact opposite. And I don't want to read both C and D, so I'm just going to circle D because it's the correct answer. Amylose has only alpha-1,4. Amylopectin has both alpha-1,4 and alpha-1,6. Okay, I've seen that question asked before. It's not the most common question because this topic is a little bit easier. It's just memorization. But it's important to recognize that amylopectin has 2 different kinds of linkages. Okay, this next one, number 11, might be a little bit of a more reasonable question your professor may ask you. What kind of glucosidase? So let's dissect that word. ACE means enzyme. Okay, I lost my mouse. There it is, okay. So, oh, I'm way too high. That's what's wrong. Okay, so ACE is enzyme. And specifically, this is about glycosidic bond. So it's the enzyme that breaks glycosidic bonds. Glycosidic bonds. Okay, what kind of glucosidase could be used to convert amylopectin to amylose? Okay, so let's think, and this is where I think it's really helpful to use my definition of amylopectin, because amylopectin is amylose plus alpha-1,6 branches, right? So if we could get rid of every alpha-1,6 bond, we would be good to go, right? And that's actually an option here, right? Alpha-1,6 glucosidase. And that's actually what ends up being correct. B is the right answer here, right? Obviously, C and D are both beta, so they don't really matter for this one. And A would end up resulting in us breaking every alpha-1,4 linkage rather than alpha-1,6, and that would be not amylose. Okay. All right. So this is the last topic for carbohydrates that I want to talk about. I don't see a ton of questions about this on exams, but I have seen them before. So I really, it's fair game. Actually, sorry, before I get into this topic, I want to mention there's a lot that I left out, okay? And the reason that I left a lot out is because different professors have very different requirements for this unit. So I encourage you all, if you're not sure, ask your professor what you need to know from this unit, because some professors, like I mentioned, will require that you memorize various aldohexoses. Some professors may say just memorize glucose, nothing else really matters. I think the most common ones are glucose, galactose, mannose, if you have to memorize them, and fructose, obviously, not obviously, but fructose. Some professors will say, no, they don't really matter. You can just look them up in real life anyways. So I don't know what they require, so I don't want to waste your time teaching you something that's not going to be relevant. And then particularly with these polysaccharides. So I only talked about amylose and amylopectin. There's also starch. There's also chitin, I believe it's pronounced. I apologize if I mispronounced that. There's, like I mentioned, glycogen. I very briefly talked about it. I didn't go into depth about it. There's cellulose. There's all kinds of different things that I skipped because they're not that important. And I don't really, I've very, very rarely seen them tested. And even then, I think it can be professor dependent. So before I jump into this topic, I want to make sure that I say, ask your professor specifically what you may need to know for this unit because I don't have that answer. Okay. All right. Now let's talk about this. So I'm sure you've likely all heard gram-positive, gram-negative bacteria, what those are, or at least like there's some difference between them. But to be honest, I didn't know what it was until I took this class. Even though I've heard it so many times, it just didn't make sense to me. Even when I looked it up, I was like, I don't really see what's different, right? So what we're going to do is we're going to look at these two diagrams. One is gram-positive, one is gram-negative, right? And we're going to look for the difference. And that's literally how I suggest doing this because I struggled a lot with figuring it out. Okay, so first thing I'm going to do is I think the right side is a little bit easier to see. It goes blue, pink, blue, cool. Okay, this one's follow a similar pattern, right? Then working our way over, it seems like it's like orange, pink, blue, orange, orange, pink, blue, orange. Okay, those are the same. Those are the same everywhere. Okay, so those are the same. Right there. That's the difference. So gram-negative, I'm actually going to undo both of those so I can highlight them. Gram-negative cell walls have just this blue linkage, whereas gram-positive cell walls, as you guys can see here, have this what's called a Pentaglycine Crosslink. Okay, so what's a Pentaglycine Crosslink? Let's break down the word. Penta, 5, glycine, glycine, and that's exactly it. So the difference here is with respect to the linkages between everything, and specifically when I say everything, I mean the N-acetylmuramic acid and the N-acetylglucosamine. So those two are connected, and they that repeats. So that's what those that's what these right here are. The blue and the pink repeating connections are that are present in cell walls, right? And so, great. So those are present in what seems to be just about from what I remember, every bacterial cell wall. However, the backbone is cross-linked. So think of it like a grid system is how I think of it, is 1, then 2, then 3, then 4, then 5, and they're connected. I guess instead of a grid system, it might be better to think of them as rungs or like as sides of a ladder. So this, and then there's another one of those exactly the same. And then just like with a ladder, we have to connect them. So this connection right here, the connection is where they differ. So in gram-negative, which I'm going to draw this gray line right here. So gram-negative is going to be below the gray line. Gram-negative, gram-positive is going to be above. Positive, okay? So, I don't know what just happened. Okay, there we go. So you have alanine, glutamate, lysine, alanine, and so on. However, the difference is, as you guys can see, actually, I don't know if you can see, let me zoom in more. this pentaglycine thing seems to come from that fourth amino acid, or that fourth circle, which is an amino acid, I should have said. And that is lysine. Okay, so lysine, for y'all who may not remember, is I'm in black. Lysine is 4 carbons, 1, 2, 3, 4. Oh, I drew way too many. So it's the R group is 1, 2, 3, 4 carbons, and then an NH2 or NH3, depending on what's it called? Depending on the pH of the area. I'm just going to draw NH2 for convenience. So this NH2 can then have, excuse me. So this is where the links happen. So that's, I should have said that's not the orange one, by the way. I apologize. That's this blue one right here is lysine. And I didn't draw the E on that. Lysine. There we go. So that's the blue one there. That can be attached directly to this alanine at this nitrogen, right? So nitrogen can, and I'm just going to draw this. Nitrogen can have methyl group. It can have a hydrogen, obviously. It can have, I think an oxygen could be directly attached fairly stably. I'm not 100% sure, but I believe so. Regardless, that's kind of very off topic. But the point is, so that's how it is in gram negative. There's a direct bond between lysine and alanine to connect the I guess the size of the ladder. That's how I think of it. That's called the cross-linking of the peptide side chains. However, in a gram positive, rather than going directly to alanine, so gram negative, I guess let's just draw right here. Oh, there we go. Okay, I lost my place. Amino acid 1, amino acid 2, lysine, alanine. We'll have this lysine bound to alanine, and then lysine, and then the other 2 amino acids, and then this lysine will be bound to alanine, and then also lysine, 2 amino acid, and so on. Over, oh goodness, hang on, I apologize. Let me zoom out. There we go. Over here, we still have those 4 amino acids. There's still a lysine. Oh, and lysine is K. I apologize, not L. I'm not going to change the ones down there, but yes, I apologize. I should have caught that. Rather than being directly bound to alanine, instead, it's got, it's got pentaglycine. So it's got glycine, 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 alanine. So glycine, glycine, and then 3 more of them, which I guess I'll just highlight in I don't know, blue. So all of these are the same. And then it attaches to the alanine, and then the next lysine, and so on. Okay, so that's the difference between a gram positive and a gram negative cell wall or bacteria. Well, bacteria, but really it's in the cell wall of the bacteria that makes it different. Okay, all right. So that is basically the end of the carbohydrate chapter in terms of the things that I think are really, really important to cover that everyone needs to know, regardless of class. However, again, your professors may have different requirements, so I would clarify with them to make sure there's not anything else. However, the carbohydrate chapter is really short and also kind of boring. So I want to move into glycolysis a little bit because there's a lot that we can talk about with glycolysis that, that'll really benefit us for the future, okay? So I'm going to start off by saying we know glycolysis is 10 reactions. I think I've said it 4 times at this point. However, glycolysis is also split into 2 phases. So the first phase, as I'm sure you guys can read right here, says the phosphorylation of glucose and conversion of 2 molecules of glyceraldehyde 3-phosphate. 2 ATPs are used to prime these reactions. That makes these are the priming reactions of glycolysis. All right, reactions of glycolysis. Right, this is the preparatory half, right? The second half is the half that gives us energy. Conversion of G3p, which I'm going to abbreviate for convenience, to pyruvate, and coupled formation of 4 ATP and 2 NADH. Okay, this is catabolism, right? So this is what makes glycolysis the catabolic process that it is. It's very important. It's why it's such a priority for our body, is this part right here, okay? So we have to do the priming in order to get that. So let's talk about the priming first, okay? So I'm just going to briefly explain the way the diagram works, and then we're going to talk about actual reactions. So the structures you obviously see, the names are to the left. If you have something along these arrows, it's usually either like energy that's formed or used in some way. Anything like the magnesium you see here is a cofactor or coenzyme that's needed. And then, so green is cofactors, coenzymes, yellow is the enzyme itself. Okay? So D-glucose is phosphorylated using hexokinase or glucokinase. So if y'all remember from the, gosh, it must have been week 6 or week 7, the time when we learned about the 6 types of enzymes, and then we went into a little bit more detail. We talked about phosphate being kind of the king of controlling physiological conditions. Well, kinases and phosphorylases add and remove phosphate groups. Okay, so hexokinase adds a phosphate group from ATP. Okay, so at this point, I'm just going to keep a running tally on the left side and then on the right side on the other half. But we are down 1 ATP. We've used 1 up. All right. Okay, so let's take a look at the next reaction. So now we have glucose 6-phosphate. Oh, I guess I should mention that. We have G6p, so glucose, and then on position 6, there's a phosphate. So if we think about the numbering, this was 1, 2, 3, 4, 5, 6. Glucose 6 has a phosphate. Makes sense. Okay, cool. So now we have phosphoglucoisomerase. Again, let's take it apart. Isomerase means something about it is going to change, but we're not adding or subtracting things. It's just going to move around a little bit, right? Phosphogluco is what it's acting on. Phosphorylated glucose. It's in the name, right? Super easy. Cool. So isomerase reactions generally don't take a lot of energy because it's just rearranging a little bit. And in fact, that's what we see here. So now we're shifting from a pyranose to a furanose. And not just from, glucopyranose to glucofuranose, excuse me. We're shifting from glucose to fructose. Okay, so the way fructose works is the numbering starts on this side, so 1, 2, 3, 4, 5, 6. Okay, and as you guys can tell, it's the same carbon. It doesn't change anything. But carbon number 6 is still phosphorylated. We've just changed its form. Why do we change its form? For the next 2 steps. Right? So now we're using phosphofructokinase, right? Once again, kinases add/remove, in this case add phosphate groups using ATP. Phosphofructo is the thing it's acting on, which is fructose that's been phosphorylated. Again, super easy, it's in the name. All right, we've used another ATP, so now we're down to ATP, right? And now let's take a look at the product. Fructose 1,6-bisphosphate, okay? So 1 and 6, once again, refer to the positions of the phosphate. And we say bisphosphate to say there's 2 of them, right? Bi is 2, bisphosphate. Okay. And once again, the numbering is 1, 2, and so on. Okay. Nothing really complicated here. This is where things get a little weird. Aldolase, all right? And it's fructose bisphosphate aldolase. And I'm really going to be talking about these 2, 4, and 5 together, all right? So an aldolase does an aldol reaction, which is a cleavage reaction, right? And in this case, it basically cleaves it right down the middle, right? And when it does that, we're left with the ketose, DHAP, and the aldose, G3p, okay? And these are actually the exact ones we talked about when we first introduced ketoses and aldoses, except they've got phosphate groups attached. That's the only difference, right? So for the sake of glycolysis, this is the one we care about. G3p, glyceraldehyde 3-phosphate. This is what we really do want. So that's why this reaction 5 is kind of included here as, like, before we even touch anything. It's a isomerase. It affects a triose, which is just a 3-carbon sugar that's got a phosphate group attached. And what it does is, so this aldose reaction gives us 1 of each of these. What it does is it converts this DHAP to G3p, so now we have 2 of it, okay? So every reaction on the right side, 2 of them happen, okay? All right, so like we mentioned, the right side is why we're doing this. This is what we care about, okay? So, it's kind of awkwardly drawn in my opinion because it doesn't draw the product again at the top. So we're taking G3p and we're using this enzyme, okay? And the enzyme that we're using is G3p Dehydrogenase, okay? So this is the thing it acts on, which is the exact thing, awesome. A dehydrogenase is going to do a redox reaction in some way. So since it's doing a redox reaction, hey, look at that. We're reducing NAD plus to NADH, and in turn, we're oxidizing G3p. And the way this oxidation works is with the help of this inorganic phosphate, we get over here 1,3-bisphosphoglycerate, okay? So if you notice, this is now no longer an aldehyde. Aldehyde, no, okay? So it's no longer called glyceraldehyde, right? So the bisphospho makes sense because there's two phosphate groups. The fact that it's not a glyceraldehyde makes sense because it's no longer an aldehyde. So it's just given the name glycerate, okay? I believe there's a functional group that like we name multiple things glycerate. I just don't know it. And to be honest, it's not super important. Okay, so the next reaction, I guess actually on the side, I need to keep a running tally. So this reaction, each time it happens, gives us one NADH. Since once again, we have two G3p, we have two NADH because we did this reaction twice, okay? Now, once again, we're dealing with a kinase. We know what kinases do. They move phosphates, right? This is phosphoglycerate kinase. We're dealing with a phosphoglycerate. Things are making sense. Awesome, okay? So now we've gained 2 ATP. So we were at -2 ATP. Now we're at +0 ATP. We've no longer lost or gained anything. This is the point at which we break even. Break even with respect to ATP. And we're going to be talking about all these reactions in a lot more depth next week. I just want to introduce them because they are really, really important. Like this is most of your exam, okay? So bisphosphoglycerate turns into phosphoglycerate because we removed one phosphate. It makes so much sense. I love it. Okay, so now the next reaction is a mutase reaction, right? And it's phosphoglycerate, whole phosphoglycerate mutase, makes sense, right? Mutase. What that word means, or not what that word means, this is a isomerase with just a weird name, basically, right? I think of it as literally just isomerase, okay? So now instead of 3-phosphoglycerate, we have 2-phosphoglycerate. Okay, makes sense if it's an isomerase or like some variation of an isomerase that just moved something around. Cool, all right. Enolase, that's the enzyme that catalyzes this next reaction. Okay, I don't know exactly what that means. Let's take a look at the product to confirm it, okay? And specifically, let's take a look at this part right here, okay? So when I think back to organic chemistry, OChem 2 toward the end, you learned what an enol is, all right? And it literally is an ene and an ol, right? So it's an alkene with an alcohol right on it. Hey, that's exactly what we have here, okay? In other words, an enolase forms an enol. And so the product that we get is phospho, because there's a phosphate group, enol, because there's an enol group, pyruvate, okay? So now we've shifted from glycerate to pyruvate because we've changed something, okay? And once again, we have cofactors, in this case, magnesium and potassium, okay? And actually, we have those same cofactors down below. So we're just gonna repeat that same process. All right, now let's talk about pyruvate kinase. So once again, we have two parts. Kinase, I think we're all pretty good on. You guys are tired of me talking about. Cool. Pyruvate. Well, it's phosphoenol pyruvate, and we get pyruvate out of it. Okay, makes sense. All right. We also form 2 ATP from this kinase. So now we've had a net total of 2 ATP. So we made 4, but 2 of those, like we used 2 to make 4, so our net total is +2. Okay, all right. So that gives us pyruvate kinase, or I'm sorry, that pyruvate kinase gives us this pyruvate. And really it gives us, like I mentioned, 2 of them, right? because we do each of these reactions twice, one with each of the G3p molecules. Okay, so we have 2 ATP. All right, so let's come back up to the top, or not to the top of the glycolysis part. So it tells us we get 2 molecules of G3p by using 2 molecules of ATP and 1 glucose. So we can literally write that out and say phase 1, glucose plus 2 ATP gives us G3p, because I don't want to write glyceraldehyde 3-phosphate, plus, 2 glyceraldehyde 3-phosphate. Actually, there's not really anything we're increasing by, right? Yeah, I don't need to say plus anything. That part is kind of irrelevant. I have lost where I was. I apologize. Give me one moment. There we are. Okay. And now phase 2 says 2 G3p plus 2, I guess I could have written ADP, but we're not going to worry about ADP for now. 2 NAD plus is going to give us 2 pyruvate plus 4 ATP plus 2 NADH. Did I miss anything? ATP, NADH. Okay, that's good. All right. So what we used, so our reactants overall are we added glucose, we used a little bit of ATP and a little bit of NAD+. And out of it, we got even more ATP than we put in, even like NADH, which we can now use because the NAD+ form has no energy for us. And we got pyruvate, which, like I mentioned above, is that most connected metabolite. So this is the big reason. Well, there are a lot of reasons why glycolysis is so important, but this is one of the big reasons that glycolysis is so valuable. Is because it gives us pyruvate that we can use to do almost anything. If we need some amino acids, use pyruvate. We need more energy, use pyruvate. We need whatever, use pyruvate. It's so handy for just about everything. That's one of the big reasons glycolysis is so valuable. Okay. So it's looking like we have about 8 minutes left. I don't want to work ahead any further than I already have. So if you guys have any questions, I'd be more than happy to take them. And if we run out of time, even then, come to drop-in tutoring. I'm available right after this session until 8 p.m. But otherwise, that's all I have for you all. I hope that helped. And yeah, see y'all next week.